I'm Aaron. I'm an independent researcher here at Uppsala University, and I sort of made the title a bit more vague, saying that I was studying horizontal gene transfer in eukaryotes. Really, it's in fungi. And so I'll give a little bit of background to that, but I think all of the methods certainly and the comparative genomic aspects will be applicable to everyone, I hope. So in particular, I want to introduce sort of the main focus that I work on, which is molds. You're all probably very familiar with these, whether you want to be or not. These are mostly, so this is the subphylum Mycotina. Mold is technically just a morphology, but we'll gloss over that for now. They're really great systems to study for genomics. They're really easy to culture in the lab. They've typically got small genomes, 40, 60 megs, with low repeat content. So that means that we can get very high quality genomes very easily. Even with Illumina, sometimes we get pretty good assemblies. We also have multiple model organisms in which to sort of benchmark a lot of our genomic results to, which is really nice. And then on top of all this, many of these fungi are important as pathogens to humans, plants, other roles. There are a lot of large-scale initiatives to sequence these. So we're talking, 1,000 genomes, 100 of which might be long read that we can start mining and looking through. So really fantastic system to do genomics in. And although when I talk about mold, you might think of this moldy lemon here on the right, the diversity of Mycotina is really, really incredible. So some of them do make mushrooms. They make fruiting bodies like morels and truffles, which are delicious. They can form ectomycorrhizal relationships with plants and can be critically important for that. They're also usually what make up part of a lichen and can be extremely diverse and resilient as this. So this lichen is growing on a rock in Antarctica. And then they also have lots of interactions with other organisms, of course. They can grow on or in plants. They like to infect insects, including the zombie ant fungus, that's Mycotina, that's got pretty famous lately. Also as ectoparasites, or even as predators, sort of actively trapping nematodes to eat them. So all of this falls within Mycotina. And it's really an astonishing amount of diversity that's sort of occurred over the last 400 or so million years. But what's also pretty incredible about this group is their ability to continue to adapt to novel environments. So we have all of these very man-made, very structured environments that we keep finding these fungi in. So we can find them, a kilometer below the earth in mines contaminated with heavy metals. We can find them contaminating specimens growing in jars of formaldehyde. We can find them in highly salt-containing saltines, and also in things like dishwashers that are important for, say, sterilizing hospitals. There's a lot of, or there's some fungi that specifically seem to have adapted to survive like 70 degrees, 80 degree warm water treatments, and now they're a big problem in hospitals. So it's not just their sort of historic diversity, but also their sort of active ability. And when I talk about this, it's not just a single species that you typically find in these novel environments, but you have it happening in different classes, maybe 3, 4 different species from 4 totally different classes will all pop up in these environments somehow. And what I'm really interested in is understanding sort of what is the genetic basis of this adaptation? How are these fungi really able to do all these sort of fantastic things? And maybe you think this is an easy question to answer. Of course, we know the genetic basis of adaptation. It's mutation, right? Easy, end of story. We can all go home. But often there's quite a bit more to it than that, which I think most people know. But let's just take one example for now. So in this case, let's look at antibiotic resistance, right? So this is a case of rapid adaptation that's occurring in the environment. Many different types of bacteria are all responding to sort of our use and our overuse of antibiotics. And it's becoming a major problem in clinics and things like this. And if we were to look at these cases and ask, how are these bacteria adapting to these novel stressors? Is it mutation? And for a couple of antibiotics, that's true. But actually, for many of them, that's not what's going on at all, right? They adapt to these novel conditions by horizontal gene transfer, right? So this is the case where instead of vertical transmission, where you either have just clonal reproduction or sexual reproduction, they're able to exchange genes between individuals, often via what's known as conjugation, sort of bacterial sex in these cases. So mutation isn't the whole story. We need to consider these other processes, and horizontal gene transfer can have very significant impacts on evolution and adaptation. And although for a long time, people really were only thinking about horizontal gene transfer in the context of prokaryotes, more and more and more, as we're getting more genomes especially, we're finding that eukaryotes do it too. So there's been a bunch of great papers recently. This one up here is from a study on grasses that found a lot of HTT in grasses. This one down here is a much more recent one on insects, finding that especially lepidopterans have huge signals of HTT. And in fungi, we've actually been seeing traces of HTT now for quite some time. And it's becoming very accepted that there's a lot of HTT going on in fungi. And particularly, I want to point out that when we see HTT in fungi, we often see entire gene clusters transferred. So in this case, the ACE1 gene cluster is important for infecting rice. And we know that the whole cluster is basically transferred from a species called Magnaportha to one called Aspergillus. So it's not just little bits and pieces of DNA or individual genes, but whole big chunks of genes that are moving around these genomes. And I really want to understand, how is this going on? And is this the secret to a lot of that diversity that I sort of presented at the beginning? A lot of people have just been sort of waving their hands and saying, well, fungi, they are osmotrophs, they suck up a lot of material from the environment, nutrients, DNA, maybe it's all accidental. But there are it's happening too often, and there are far too many coincidences that I wasn't satisfied with that answer. And I could sort of tell the story that way, as though I had this hypothesis and a specific idea and chased it down, but that's not how it happened. And I think it might be nice for people to see the real story. So we'll take a slight detour through what I was doing my postdoc on. And this was a really fun project on meiotic drive in fungi, right? So these are sort of selfish genes that cheat Mendelian ratios in order to bias their proportions in future generations. So we're looking at the sexual spores of a specific fungus here called Prospera anserina. And normally it produces 4 spores per ascus, and that's the 4 products of meiosis all packaged into a spore, essentially. But if you look over in the B panel, you can see that some of the asci only have 2 spores. And what's gone on here is that these 2 spores carry spore killer genes, and they've actually killed their other 2 sibling spores. So this is really interesting evolutionary scenarios. Meiotic drive is found in all kinds of organisms, so we wanted to understand, how is this happening? And so to do that, we've generated a lot of high quality, telomere to telomere assemblies of these fungi, and we had a clear phenotype, and we just started mapping, what is doing this? Where are these spore killing genes? And the results sort of shocked us. So what I'm showing here are 3 strains with different spore killing phenotypes, and I've just aligned all of chromosome 5. And these strains are extremely similar. They're more than 99.99% identical in protein coding regions, almost clones. But then they have this huge chunk of DNA here highlighted in blue that can be on sort of one side of the chromosome, the other side of the chromosome, or totally absent, or it's not shown here, is actually there's 2 other chromosomes that we found it on. And of course, this is where it turned out our spore killing genes were, so SPOC for spore killing. And so this was behind our phenotype, which was really great. And we wrote that paper up, sort of somewhat the end of that story. But of course, it left this big question of how on earth is this giant block of DNA moving around between otherwise, basically identical strains. And what we could do, since we had many genomes, we could just align those genomes and look at the insertion site, look at the boundary. And so in this case, what we're talking about is 250 kb of sequence. That's how big this SPOC block is, so it's all cut out here. But we're looking at exactly where it sort of jumped in this genome. And it aligns very, very cleanly. There's no messiness, there's no weird breaks, nothing like that. And you also have this very small motif sort of on either side. And actually every single block, so this is just for one version, but like I said, we have multiple strains with multiple phenotypes. They all have different blocks in different locations. They all look exactly like this. Very clean insertions at exactly this same motif, which was super suspicious. And we brought it to Alex actually and said, Alex, what is this? And he said, oh, well, maybe it's a transposon. And so I'll give a very, very brief background for anybody that doesn't think about transposons a lot. So basically, they typically come in 2 flavors. There's the retrotransposons that use an RNA intermediate, and the DNA transposons that sort of go directly cut and paste DNA to DNA. in one way or another. And in particular, I just want to point out these things called kryptons that don't have the typical features of other DNA transposons. There's no big tears at the ends, things like that. And instead of an integrase, they have a tyrosine recombinase. They're also target site specific. But these things are normal TE size. They're 2, 3, maybe 4 kb at the most. They're not 250 kb. But sure enough, when we dug into this region containing our Spock genes, the Spock block as we called it, we found a putative tyrosine recombinase that we ended up naming Kirk. And so we figured out that this whole block is probably acting kind of like a krypton and transposing around the genome. So we more or less renamed it to Enterprise, this sort of giant transposon in this fungus, which was great. And I wanted to know, is this some weird, strange, unique TE that somehow got massive? Was this thing, is the ancestor of this thing, only 2 kb and we were looking at some strange giant bloated one? Or is there something else going on? And looking through the literature, there was only one other example, but it was also big. It was about 70 kb. So that was pretty promising. But I kind of got stuck there. I couldn't find anything else. I was searching around genomes for other big things, and I kept hitting walls. Partly because a lot of the genomes I was looking in at the time weren't very high quality. I wasn't getting telomere to telomere at that point. And I just couldn't figure out what was happening. And so after sort of 2 or 3 years of really trying to dig into this, I ended up collaborating with a guy who's sort of an expert at studying gene clusters, and he had some really nice genomes. His name's Emil, and he helped me really crack the code. And we discovered the starships. So this whole family of giant mobile elements, a lot like the one we saw in Perospora, widespread all across Mycotina, and really huge range in their size. So the smallest ones are about 25 kb, the biggest ones are over 500. And a lot of what that extra genetic makeup is what we call cargo genes, right? So the actual tyrosine recombinase, that's normal size, it's like 2 kb. The rest is all a lot of other fungal genes that do many, many other different things. And we find this in many different types of fungi. So the molds, the filamentous fungi, right? A lot of pathogens have it, lichens have it, as well as a lot of symbiotic fungi typically have it. So you name it, these things are in those genomes. But what exactly are they? Why are these giant transposons hanging out in these fungi? And why are they picking up all these fungal genes instead of just their own machinery to replicate themselves? What is going on here? And if we look more broadly across systems about what these look like, we have something that looks exactly like this, but they're in bacteria. And these are integrative and conjugative elements, right? So these are the plasmids or the integrative elements that bacteria use to share genes among each other. This is exactly how bacteria do horizontal gene transfer and adapt to their environment with these mechanisms. But I'm sure, if fungi were making these weird structures to trade genes, we probably would have noticed way before this. So we don't think that's what's happening. But if you look at other types of bacteria, and specifically actinomycetes, which are the filamentous bacteria, they don't make this pilus structure. They actually exchange their ICEs with hyphal fusion. And of course, fungi have hyphae, and fungi do hyphal fusion. And actually, we already know that fungal hyphae can exchange plasmids through fusion, and even whole chromosomes through fusion. So the model here is that the starships are really fungal analogs of these bacterial integrative and conjugative elements, and that their role is really to trade genes among unrelated strains in very much the same way that bacteria use HTT to adapt to their environment. Okay, so we wanted to understand these things better. This is all sort of from bioinformatic inference of what we were able to glean, but we wanted to know the biology of the starships a lot better. And so we needed a model system to do this in. And this is where Andrew Urquhart came in, studying the only other one in the literature that I could find that I sort of alluded to, that he had named Hephaestus. And Hephaestus is pretty cool. It's a bit smaller than Enterprise. It's only about 70 kb. It's in this species of fungus called Paecilomyces variatii, which typically contaminates grain in storage, so it's a problem. And he found that the genes it carries allows this fungi to resist up to 5 different heavy metals. So lead, arsenic, zinc, cadmium, mercury. It's able to provide resistance to all of those stressors in one single sort of giant mobile gene cluster, which is pretty cool. Paecilomyces is also very amenable to genetic manipulation, and Andrew had all the skills to do that. So he started digging into how this thing is actually moving around and interacting with the genome. And so the first thing he wanted to know is this actually able to move? Do we see this sort of large polymorphism and see it in different places because the whole thing is transposing? And yes, it is. They're definitely mobile. So he set up a really elegant experiment where he took Hephaestus, right, this whole 70 kb element, and he put hygromycin right next to it on one side in the genome. But he put the promoter for it on the complete other side. So normally the promoter and hygromycin are 70 kb apart. You're not going to get hygromycin expressed. But if Hephaestus jumps out, then it brings the two ends together and you get hygromycin expressed. And that's exactly what he saw. So that's sort of in this center panel here. The control is the top left. The rest are all derivatives. And you see normally it's not resistant to hygromycin. But in all these cases, it excites itself out and you get growth. And then what's great about Hephaestus is it sort of carries its own reporter genes because it has this ability to resist heavy metals. So we can also assay it on cadmium and zinc. And you see that most of the derivatives still have it. So in all but 2 cases, it's actually reinserted somewhere else in the genome. So yes, it's able to jump around in the genome, which was really great to see. He then went a step further and constructed what he calls mini Hephaestus, where he just sort of took the beginning, including the captain gene, the tyrosine recombinase, the very end, and then that zinc reporter gene. And he put that in the genome so that he could sort of monitor the movement at a higher throughput. And that's exactly what he was able to do. It still works. So that sort of shows that this minimal component still has all of the basic needs to jump around, kind of helping to confirm that it is this captain gene that's moving this. He took it a step further and then did site-directed mutagenesis of basically exactly the residues that we would expect to be important if this was a tyrosine recombinase, by checking it to other well-described tyrosine recombinases. So here's sort of a count of how often mini Hephaestus normally jumps. You do a frame shift and you completely lose any movement. And it's sort of important that it's a frame shift and not a knockout, because you could imagine a scenario where there was some host gene, something in the fungal background, finding this region and chopping it out and moving it. But if it was just a frame shift, it should still be able to find that sequence. The only thing that we've broken is that protein. And so this really shows that it is that gene that is moving this cluster. And then, of course, he was also able to rescue that in trans as well. So yes, this is a TE. It moves by tyrosine recombinase, just like some of these other systems, including these bacterial systems. He was also able to confirm that it's site-specific. So this is something that we had sort of seen from the genome, but now we could really scale up. So in this case, we looked at 123 different insertions across these mini Hephaestus moving around, and confirm that it specifically does target TTAC. And there's also a conserved A that's about 10 base pairs downstream in order to insert. It turns out we have many Hephaestus elements, sort of the Hephaestus family, we call it now, spread across this group of fungi. And if we look at the boundaries there to see where they're inserting, it seems like it's an evolutionary conserved site. So all of these elements actually seem to target this TTAC, which is pretty interesting for us. So that's all sort of about the integrative aspects. Can this move around a genome and insert itself in and out? The conjugative aspect is, can these things actually move between individuals, right? And so in this case, we got lucky that we have, again, a pretty good wealth of genomes to look at. And in this case, what this tree is showing is sort of the focal species, Paecilomyces vireotei, where again, Hephaestus is polymorphic. It's in some, but not others. And then it has the sister species, a couple of them, but one in particular called Paecilomyces lascivitis, where one of the species, or one of the strains, seems to specifically have Hephaestus and none of the others do. And we wanted to know if that was because it transferred horizontally. And you can't really take a phylogenetic approach to evaluate this because, of course, they're sister species, and this is a present absence variation, so you can't just build a tree. So instead, he did a best blast approach. And he just chopped the genome up into a bunch of bins and then looked at the best blast reciprocal hits and see how they fell out. And you can see sort of that they form a nice little distribution around 80, 85%. So even though they're in the same genus, these are two very different species. And then they have this weird peak right at 100%. And if you take all of those bins and just map them back to the genome, surprise, surprise, everything, all the red bins, that's Hephaestus. And so that thing is 100%. It's definitely not part of this regular background. And what also popped up is an even bigger region in blue that all maps to the same region. And it turned out that was a new starship we didn't know about named Pegasus. So both of these elements went horizontally from Paecilomyces variotii into Lepidopteris. I did it very recently. And they actually represent, if you sort of add up all of their base pairs compared to the whole genome, that's about 1% of this fungus's genome that recently came in from HGT. So we think that these are, being like bacterial ICE elements, they can integrate, they can conjugate, they're doing everything you need. So can they actually drive rapid adaptation, right? Like if we're getting back to what I proposed at the beginning, where these novel phenotypes that we see arriving are actually being driven by something, is it the starships? And I'll give sort of two quick examples that I think really illustrates well that yes, they can do this. And so the first one is a story about a wheat pathogen. So in 1941, something sort of terrible happened where this brand new disease started affecting a lot of farmers' fields, and it rapidly became a global problem. And it was causing these sort of tan spots on the leaves, so it got the name tan spot disease, very creative. And it turned out it was caused by Pyrenophora tritici. And this was never known to cause disease in wheat before this. And already quite a long time ago, they figured out what happened was that this very small effector gene, so effector genes are sort of the ammunition that fungi typically use to infect plants, called tox-A, it was actually transferred from another fungus called Parastegnosospora nodrum through horizontal gene transfer. And it was pretty clear about that. But that's as far as that story went for about 20 years, 15 years, I suppose. And of course, I'm telling you this in this case because, yes, once we figured all this out, we found that tox-A is actually located on a starship. And that starship's named Horizon. If you actually look at different genomes of Pyrenophora, that starship is in different places, which means it's actually still moving around, it's still mobile, and therefore can still transfer to new fungi and continue to basically allow new species of fungi to infect the most widely cultivated strain of wheat, which is a problem. The next example I really like is, I think, really hits home this sort of story of convergent evolution being driven by these starships. And in this case, the phenotype that we're looking at is formaldehyde resistance. So things should not grow in formaldehyde. We specifically use it as a preservative to stop things from growing. But we found some starships that look like they carry genes for resistance to formaldehyde. And of course, our focal species here, again, Paecilomyces variotii, had a starship we named Galactica, and had sort of a clear gene cluster that looked like it was for formaldehyde resistance. And when we checked it out, so here's a bunch of strains growing on different concentrations of formaldehyde. The ones with Galactica are able to tolerate really, really high levels of formaldehyde compared to the ones that don't. So this is providing a huge benefit for these fungi in this environment. And already we sort of saw that Galactica had some signatures of horizontal gene transfer, but when we specifically chased down this gene cluster, the story got even more interesting because we found it in two completely different starships, totally unrelated to Galactica, one that we named Mithridate and one that we named Prometheus. And you can sort of align, these are the genes in the cluster. It looks like Mithridate probably independently acquired the cluster at a different evolutionary point in time. This should say Galactica. But we have some good evidence that Prometheus actually stole the cluster from Mithridate, and I can talk about that later if people are interested how we know the direction. And with Prometheus, what we can see is that it's then undergone multiple HGT events very rapidly. So it seemed like Prometheus wasn't really doing much, was probably mostly undergoing vertical descent. And then we think in Aspergillus sidowie, it picked up the formaldehyde resistance cluster by stealing it from Mithridate. And then we see rapid HGT occurring at many different orders of sort of phylogenetic distance. So we see it jumping to other Aspergillus species. We see it jumping between families to a strain of Paecilomyces variotii, a different one than the ones I've been talking about. Interestingly, this one was found growing on the walls in Chernobyl, so that's pretty cool. And then we also see it jumping even between orders. And if you look at Prometheus and all of these 4 species, it's basically identical. This is clearly happening like right now. So yeah, the way that we're sort of putting this together as driving sort of convergent evolution of these phenotypes is that although generally the starships seem to sort of stay in their focal species, but if they manage to pick up these adaptive genes or adaptive gene clusters, then all of a sudden selection sort of benefits their spreading, and we really see a burst of HTT events. Another point that I don't have so much time to talk about is that we actually often see sort of the starship components, and specifically the captain, start to degrade rapidly. So potentially if these things happened in the past, we couldn't see them. So these could be moving genes and gene clusters around, and then sort of the signatures of the starship can disappear, which actually makes their true impact pretty difficult to quantify. So, right, so how do we do that? How do we start getting at the actual impact of these things? How do we understand these things a bit deeper? And we're going to go deeper and deeper and deeper here. And looking at these sort of individual case scenarios of individual ships and individual species is pretty limiting. So we need to be able to do this on a broader scale. And so we've been developing a pipeline in order to automate a lot of the starship identification and curation, which is our current major challenge. And this is a lot of the work from my collaborator, Emil, again, who's been a lot of the great insights from this. And he's named it Starfish. And I won't go into the details so much right now, but essentially this takes a comparative approach in order to identify these regions. So more or less, you identify large indels between closely related strains or species and see which of those line up with protein domains that we know are of interest, so the ones that we typically find in other starships, and then tries to predict their flanking sequences and has some nice visualizations as well. And we've been testing this, and so far it seems to be very successful. And so one of the databases that we're sort of using as proving grounds is this collection of 524 Aspergillus fumigatus genomes. And this is an important fungus. It causes a lot of human disease. So there's a lot of genomic resources for it, and people are also very interested in it. And some of these are long read. I think 11 of them are long reads, and the rest are all short reads. So from this population now, we actually pulled out 20 high confidence starships. So there might actually be a few more. But so we're not just talking about 1 or 2 per species now, like this seems to be what's going on at the population level. But actually most are pretty rare. So all of these are, the dotted line here is the 5% line. So many of them are pretty rare in the population with just a couple that are common. Nevertheless, a lot of these rare ones have very interesting genes, things for biofilm formation, things for arsenic resistance, different biosynthetic gene clusters. So even if they're not common, they're still doing important things and actually really shifts our perspective of how much of the genome is mobile than we had ever really appreciated beforehand. And I want to highlight one in particular because I think it gives really interesting insights into sort of another aspect of these giant TEs. And this is in regards to fumigermin. And so fumigermin is produced by Aspergillus fumigatus, but only in the presence of Streptomyces rapamycinosus, right? So this is a strain that produces rapamycin, an antifungal. And you can see if you grow the fungus on its own, it's not produced. If you grow it with other Streptomyces, you don't really get it. But specifically with that species, you really see this compound produced. And of course, I'm talking about it because it turned out from our analyses that's actually in a starship that we named Lamia. And what's pretty interesting to us is that the original paper that sort of found it could only find it intact in a single strain. So in fact, in every other strain they looked at, it had accumulated point mutations or deletions and was non-functional. And the active form was quite rare. And so what we think is going on here is that Aspergillus fumigatus actually undergoes facultative sex. So it has a clonal life cycle and then sometimes does sex. And if you have a gene or a gene cluster that isn't always expressed, if it's undergoing clonal replication when this gene isn't expressed, it can accumulate mutations. And potentially some, from Muller's ratchet, this type of stuff, that gene gets eliminated. And if that were the whole story, it would probably lose that gene and not be able to competitively interact with the Streptomyces species anymore, right? It would be less fit, probably wouldn't be able to invade those environments. But as long as there's any Aspergillus fumigatus strain there, it can just pick it up again from one of its conspecifics in the environment, and away it goes. And I think this is something to think about more broadly as well, because it's not just fungi that do facultative sex. There's a lot of animals and plants that also do facultative sex. And they're also going to be susceptible to these types of dynamics. And so getting towards the end here, just to sort of summarize and leave a couple more bits and pieces for you all to think about. We've discovered the starships. They're this family of massive mobile elements that are prevalent across fungal genomes, specifically the Posiziomycotina. We think we've provided pretty good evidence that they're analogs to prokaryotic ICEs, integrative and conjugative elements, and that they can move around both within genomes and move between individuals and individual genomes. We've shown in at least a few cases that they do drive adaptation, and in many cases rapid adaptation to novel environments. And we think that this is important on a sort of broader evolutionary timescale for maintaining very flexible lifestyles, so that many of these species can live in soils or live on plants or live in human environments just by swapping out these genes in these starships. And so if we circle back to the question I asked way at the beginning of what is the genetic basis of adaptation, at least in these fungi, I think it's the starships and horizontal gene transfer. And I don't think this is just going to be the case for fungi. As I mentioned at the beginning, HGT is starting to be appreciated in a lot of different eukaryotes. And I think we already have another case of this, and I think it's in fish. So there's this element called teratorn. It's also large, it's 230 or so kb, and it's a mobile element that's actually derived from the fusion of a piggyback transposon and a herpes virus genome. And this is apparently widespread across teleost fish. When you look at the contents of this, it's not just herpes and piggyback, there's actually fish genes in there too, meaning that it could mediate HTT among different individuals, different fish, different species, who knows? It hasn't been dug into too much. There's also compelling evidence that the grass scenario could be some sort of mobile element, also things from Cominomonas. So I think we're going to start finding giant TEs in more eukaryotes and really have to start appreciating the role that these have on adaptation and evolution. And so with that, I have a bunch of people to thank. My lab here in Uppsala, Emil, who is my collaborator, who cracked the code on a lot of this stuff and really has done a bunch of really cool stuff on this. He's starting his lab in Madison, Wisconsin, and is looking for good postdocs and PhD students, by the way. Also, Andrew, who recently started a postdoc with me, did a lot of the work I presented as well. And then some key collaborators, including, of course, Alex, and then as well some Swedish funding, where all the money came from. And yeah, thanks for inviting me here. Happy to take any questions or discuss further.